When we look at the regulators, which we're talking about all of those different hormones that I described on the video that are being sent out, they come from the nervous system and signals from the nervous system. Your brain makes a decision whether it's conscious or not. And then what happens is small molecules are sent out, or hormones are sent out from a couple of different tissue types. And what we're going to get is then amplification at different tissues within the body to send out different types of signaling pathways to control metabolism. And so we're going to look at how these are connected. And you can see if our brain is going to start the process, the hypothalamus and the pituitary gland being right next to them seem to be the first in the line of those things that are stimulated. And so we get sensory input from our brains, whether that be consciously or unconsciously, and it will interpret whatever the signals it sees. Now, most of the signaling processes occur through the hypothalamus. Now, you see a little arrow that comes way over, all right, and it kind of bypasses. The production of epinephrine needs to be very, very quick, and we talked about how overarching the epinephrine actually is in regulating lots of different tissue types. And so the central nervous system, if you do need a very quick signaling, we get epinephrine up and running. All right. The other types of inputs that actually give out signals is the talk about insulin, glucagon. That actually is indicated by the blood glucose level, which is not involved from the central nervous system. And the pancreas is the one that's detecting it. And so we can have control. But everything else is directed from the central nervous system. Now, the hypothalamus will then send out small hormones and the hypothalamic hormones, and then they will affect the other targets. Now, there are first targets and second targets. All right. The hypothalamus will send out items that will affect either the anterior or the posterior pituitary gland, and depending upon what the signal is, it will send out different signals. Now, if you take a look at all of these in terms of the pituitary gland, you can see an M in front of it, and so they are all either small proteins or small peptides. So they are actual proteins being sent out as the signaling molecules. So the first target is the pituitary gland. The second target are the other types of tissue types that can give off hormone signals, like the thyroid, the adrenal cortex, and then the sex organs, which then give out particular types of hormones. And probably if you actually take a look at this, testosterone, progesterone, estrogen, cortisol, these are the ones you probably have heard about at some point in time. And there's a reason why we've heard about these, and a reason why you probably haven't heard about a lot of these. I'll identify that in just a couple slides. This one. All right, central nervous system will upregulate the hypothalamus. Now, what happens is you release a hormone, but you only release nanogram levels. Okay, that is nanogram levels in your bloodstream. Now, if you take a look, nanogram levels are not that bad because you are releasing it from the hypothalamus into either the pituitary gland. So it has to travel from here to there. I don't need a lot, okay? And I can get a signal. But the problem is with those types of hormones, I don't actually go in the middle of the brain and extract blood from right here to detect what's going on. And so if you have 5 to 6 liters of blood and you only have a handful of nanograms of material in that, the concentrations are so small that it's hard for us to detect. We may not even know all of the different hormones because we haven't been able to detect the kinds of hormones. Because for us to detect it, we take it out of the blood, but once it gets past here, it gets diluted. And so it's very hard for us to detect those. Now, the pituitary gland is going to then secrete things. And remember, you either have the anterior or the posterior. And so even some of these different hormones actually have to travel around to come over and to stimulate the posterior. You get levels that are produced in the microgram region. And again, micrograms diluted all the way up to 5 or 6 liters. And then you get to the secondary target, the adrenal gland, and then we get to something that you're familiar with. At this moment in time, the concentrations or the amounts are milligram. And so if you take a look at that, nanogram to microgram, 1,000-fold, and then microgram to milligram, another 1,000-fold. And so the first hormones are 1/1,000,000th the concentration that the kinds of hormones that we're familiar with, the ones that we have names. So it's much, much smaller in concentration. Of course, cortisol is high enough in concentration that we detected that very early. And the ones that are coming off the secondary glands are the ones that we understand the most about because we discovered them first and have been studying them for a long time. The last item that we're going to look at is diabetes, and there are a few types in terms of the diabetes. Type 1, usually juvenile, and it is insulin dependent, meaning I can treat the patient with insulin. And so basically in this particular type of disease, insulin is not produced in sufficient amounts from absolutely zero to very, very little. All right. Type 2 diabetes, which is becoming a much larger problem, I usually talk about adult onset, although the ages of the development of type 2 diabetes is now not just in adults, but is becoming younger. This is some sort of insulin resistance, okay? It's usually coupled with weight gain, and it's usually a fact of you get a desensitization of the insulin receptor, and or usually it's an issue with the receptor. And so if you take a look at what should be going on, normal response in terms of glucose, okay, glucose gets transported. Insulin will then bind to the receptor. The receptor will cause us to make sure that the glucose is being transported and then processing what happens in the glucose. Type 1 diabetes, we've got lots and lots of glucose, but we have no insulin at all. And so I can't upregulate the receptor, I can't upregulate the transporter, and we get no glucose inside, and we always have high blood glucose levels. Here in type 2, the traditional way that we think about it is you have both very high levels of insulin, you have high levels of sugar. The insulin may bind to the receptor, but will actually not cause the signal transduction that is expected, and so the transporters will not be upregulated, and the processes to process glucose are not run. All right, why in the world does someone become insulin resistant? Okay, there is a lot of different mechanisms proposed. Okay, but here is one of them. And if we go back and look at the normal pathway, so we've got the insulin binding, the receptor dimerizes, I get autophosphorylation between the two subunits. That causes IRS1 to be recruited and bind, which then gets phosphorylated. The insulin receptor substrate is the first item in the pathway to be phosphorylated, which will then start doing a lot of different things: glucose uptake and glycogen synthesis, also cell division. There's a lot of things that are signaled by that particular pathway. The phosphorylation event by the insulin receptor is at a particular tyrosine that causes all of that particular process to occur. That's the normal. That's what we learned originally. Okay. One thing about being insulin resistant, even though you have sugar in the blood, the sugar is not getting into the cells, so the cells feel like they're under starvation. So in that case, you start mobilizing fat stores. One indication of a person that has become diabetic and they didn't know about it is, one, they have to urinate a lot. It's because of all the sugar in the bloodstream starts to get filtered out, and as that sugar comes out with all those hydroxides, you bring a lot more water, and so you have to urinate. And so that's not a test anywhere. Somebody could recognize that themselves. The second thing is they actually start to lose weight. And what's happening is the body feels like it's under starvation, so it starts breaking down fat. And fat can be transported into the cells, and you start using the stores of their fat. Now, as fat gets inside the cell, what happens is, or what can happen is, you can get an upregulation of protein kinase C, which then will, very weakly, but does, bind to the IRS1 protein, but it phosphorylates it in a different position, the serine. Now, what happens when that is phosphorylated at the serine? It can't bind to the insulin substrate anymore. And so if it can't bind to the insulin substrate, be phosphorylated in this position, it can't upregulate the normal processes. What it does is it inhibits a different pathway. And so because of this free fat, we get a hijacking of what the IRS1 should be doing. Okay? And so this is one of the proposed mechanisms about why this might happen. And in a very poor diet, you can have lots of fats coming out without actually breaking down your own lipid stores. You can actually overwhelm the system just with the fats that are coming into the system. All right, symptoms: high glucose in blood. Then if you ever go to the doctor and have a urine test done, they actually test for how much sugar is there. You either have low levels of insulin in one type, or you have very high insulin concentrations, but it's not processed. You have high levels of glucagon because your body feels like it's under starvation, and glycolysis is inhibited. You have gluconeogenesis going on, and glycogen breakdown is stimulated. You feel hungry all the time, okay, because you're sending out hormonal signals that you need to intake, and you become thirsty because you're excreting very polar molecules. There are a lot of treatments that are given in conjunction with insulin itself, and treatments include stimulators. One that is used quite often is metformin, and it elevates concentrations of cyclic AMP. And so cyclic AMP, not cyclic AMP, but AMP itself. And if you upregulate AMP, then you start other signaling pathways, which then upregulates and helps glucose uptake in muscle cell and fatty acid oxidation. That in the liver, it usually slows down gluconeogenesis, fat synthesis, and cholesterol synthesis. In adipose tissue, it starts to make the fatty acid synthesis go down. And in heart, fatty acid oxidation goes up, which is the normal way that it extracts energy. and it will upregulate glucose and glycolysis. And so metformin is a very highly nitrogen-containing molecule, and so a lot of people will be prescribed that. They will even be prescribed the metformin usually before you actually become insulin dependent. And so if you're in the pre-diabetic state, metformin's one of the first drugs that is used. Now, I'll tell you why or how you can find if you're in a diabetic state or pre-diabetic state in just a couple slides. What you tend to have for a diabetic is lots of glucose, but it's not in the tissues, it's all in the bloodstream. So energy is starting to derive from fats and amino acids, and what really becomes available for use around the body is what they say abundance of acetyl-CoA. What that really means is the ketone bodies are being produced, and they get produced in higher levels than normal. Now, ketone bodies are fine. The muscle can use ketone bodies. The heart can use ketone bodies. The brain is what's have a challenge with the ketone bodies. But you get them so high that they can break down in a non-enzymatic way, and this is where you can get the ketone. And we saw that in the ketone bodies. Non-enzymatically, you get acetone. And so one way to know if someone is in compliance when they're treating themselves with insulin, if they come in and you smell ketone, or the smell of the acetone, most people talk about it as a fruity smell, but if you've taken organic chemistry, you know what acetone smells like. You can detect that. It can be off their breath or even being excreted through their sweat. The downside of the ketone bodies, if you take a look at the structures of all the ketone bodies, they're acids. And so as you put more and more acids in the blood, we start to get acidosis. Your blood chemistry becomes more acidic, which then tends to excrete bicarbonate, or the CO2, which then makes it more basic, and that will disrupt all the normal processes that are optimized for the 7.4 pH, and so this tends to drive the pH in the wrong direction. Now, diabetic patients know that they're going to be tested for their insulin. Well, not really, they don't get tested for their insulin level. What they usually get tested is their blood glucose levels. And so a lot of times a patient will make sure, just like before you go to the dentist, what do? You brush your teeth, right? It doesn't matter you haven't flossed for 6 months since the last time you saw them. You brush your teeth really, really well. Well, a diabetic will also make sure that, oh, I'm going to the doctor, I need to make sure my insulin's good and my blood sugar level is great every time they walk in. Here's the problem. The past 2 months, they haven't been checking it and their blood glucose has been really, really high. And so that one check is okay, and if they're not good at that, that's a good indicator that they're not doing really, really well. But we do another test called the AC1 test. Now, what happens with the AC1 test is that the hemoglobin is actually tested, and the beta chains of the hemoglobin will actually react with glucose through a Schiff's base intermediate within a rearrangement to make a permanent linkage between the protein and the sugar. That's non-reversible, essentially. Now, you can see it is a reversible, but a little tiny arrow going backwards. If you look at an average person's concentration of sugar linked to the beta linkages, you should be in a range that is under 5% of all of your hemoglobin attached. The only time this reaction happens is when the concentration of glucose is very, very high. And so normally you eat, it does get high spikes, but comes down pretty quickly because you have the hormonal response to that, and then sugar gets mobilized into all of the tissue types. If a diabetic has a very high concentration of glucose for a long period of time, then this reaction increases. And so once you get into a numerical value of 6%, you're at the edge, and then, or really 5.5, and you get higher. a diabetic can be in the 10s and 12s%. And what this does is you might have a perfect blood glucose level when you go into the doctor, but your AC1 is really, really high. That meant over long periods of time, months, your blood sugar has not been controlled. And so this is something that they look at. And so your A1C will always be taken when you do normal blood tests. And when they look at that, they look for the percentage. If you get into a zone that becomes higher and higher and higher, you get what's termed pre-diabetic, and that's when metformin is usually used before you become insulin dependent. Once you start taking insulin, most of the time there's no reversing from that once you start supplementing, and so they don't want you to get to that point if at all possible. Insulin was first isolated and developed and given out from porcine sources. So what they would do is, from the slaughtering of the pigs to make your sausage, they would take the pancreas, the pancreases, and they would isolate the small protein insulin, and that's what they would sell. We are now at a point that most insulin is not porcine, although you can still get that. We actually prescribe mostly human insulin. So how in the world did we get that? Now we take and do cellular extractions. And so we've isolated the DNA from the human source and we produce it that way. That doesn't always mean insulin is in high abundance. Dr. West was doing some research using some insulin. When did you do that research with insulin that you couldn't get it? Was it 2 summers ago, 3 summers ago? Yeah. Yeah, in which there was just none. Yeah. Yeah, there was just none in the human supply. I mean, none in the supply for at least scientific. That also shrunk the amount of insulin that was able to be used for treatment of patients, drove up costs during that particular time. And so there is a large demand on insulin use. And so it's not always readily available like you might think. Yeah, you can get diabetics, the percentage of the hemoglobin goes higher because they've had higher or longer times exposed to higher concentrations of glucose.